and Justice Mohan sir, Gopinath sir, Punin Kana sir, and Ramesh sir, and all my friends. This is a project on phytochemical studies of Pterocarpus santalinus bark for its potential utilization. Myself is PI. I am working on wood processing division IWST, and Dr. S.R. Sukla is working in this project in wood property division, and Muthu Kumar, he is a plant pathologist. So he is also associated in this project. So I will skip this general introduction, directly come to the objective. These are the basically, since morning we are discussing with the therapeutic activity. But now in this project, we are evaluating the bark for its potential utilization on application of wood. See, there is two component of this project. On the one hand, there is bark. This is not till now. This is not any reported economical value. And on the other hand, there is wood of mango wood or rubber wood and Milia dubia wood, which is perishable in nature, less durable, and easily available. So, taking extractive from the bark of red sanders and its application on these low durable wood for the making good in appearance and durability. So these are the objectives, basically to optimize the standardization of process parameter for the impregnation of extracts into rubber wood, mango wood, and Milia dubia wood. There is 3 wood species we are working, and to optimize the retention of bark extract impregnated wood by varying the process parameters, and to evaluate the impact of application of bark extract on wood of selected species and its performance on appearance and durability against fungal attack. This slide, you can move from here. Yeah, this is the methodology. The collection, processing of raw material, this bark, and sample preparation, extraction by cold and hot, by chocolate method, determination of extractive value in cold and hot method, the chromatographic fingerprinting using TLC and HPLC, and then extract impregnation in rubber wood, Milia wood, and mango wood. For that, there is a standard, Indian standard. This is a small wood blocks measuring 20 mm cube that is impregnated with the red sander bark using full cell process, and treating solution of extract of different concentration 2 to 10%, and the impregnation, and finally the comparing the efficacy of the treatment. For the fungal decay test, we have chosen this one brown rot fungi, that is Oligoporus placentus, and white rot, the Tremitis hirusta fungi. And weight loss of treated test sample was observed in comparison to the control, and the decay test adopting agar block method was carried out as per the standard procedure. This is red sander bark. This is physical progress of last 1 year, since 2 years already happened. So this was the physical progress of the first 1 year. I will not go through deep inside. So just you can see the extract yield also, because it vary a lot. Whenever I'm first 1 kg bark was kindly provided by the Andhra Pradesh Forest Department, Tirupati Forest Department, and then after 15 kg bark was provided. Here I would like to show that in first 1 kg, these were the extract yield. And the second, which was given in 15 kg, the methanol extract was 3%. So you can see the difference. Now the physical progress of last 1 year, the procedure for extraction of bark was optimized, the chromatographic TLC and HPLC fingerprinting was done, And we have also carried out the similarity between red sanded wood and bark also, because the objective of the project is to apply the bark extractive on low durable wood. So there will be a confusion whether it has been treated with the bark or not. So we have compared with the wood also. So I will show later on the HPLC profile of the wood and bark. So and apply on the this wood surface. So coating, apart from the project objective, we have also carried out this coating of red sanded bark extractive on wood surface, and its anti-UV efficacy was determined. Anti-UV means accelerated weathering. In accelerated weathering we have carried out, in which 250 hour or 500 hour continuous UV light was exposed on the treated sample in order to see whether it will survive or in the sunlight or open condition or not. So I will come later on the results. One on the antifungal activity, one paper was presented in this conference, and we are in the process of writing a research paper also, but it will take 2-3 months. This is the impregnation experiment we have carried out according to Indian Standard 401. There is a specific standard for the wood impregnation. So in this first we place the wood block 20 cm cube, and we apply vacuum. Then vacuum so that the impregnation can happen in a better and a faster way. After that, we have a pressure. With pressure, the extract was impregnated into. You can see this. This is extract as well as wood, and we close it, and we have applied the pressure for 60 minutes and 120 minutes. This is the treatment. This is after treatment. Wood treated with methanol extract, wood treated with acetone extract. This is methanol plus acetone extract, and this is water extract. So these are the different extracts. So extract was used for impregnation. 3% water extract was used, 3% methanol, and acetone extract, 3% methanol acetone, 4:1. It was extracted in the mixture. And treatment procedure was full cell procedure was used to treat rubber wood, also for Melia dubia and mango wood, because in first set we have treated the rubber wood. This was done in 3 simple steps. Initial vacuum 500 to 600 mm, then pressure of 100 to 120 psi, and final vacuum was 500 to 530 mm for 3-5 minutes. Then after treatment, the sample was conditioned at room temperature, and final weight was measured. And depending upon the extractive, we have come with the water extract was impregnated 3.2 kg per meter cube of wood. 1 meter cube can, by this method, 3.21 kg of extractive is required for 1 meter cube for wood. So similarly, the case of acetone extract, methanol extract, and that. So this is the weight gain percentage. This is the dry weight gain percentage. This is the dry weight retention. So dry retention is 3.21. For water, 3. Like that, 3.38 for acetone, and like that. Mango wood impregnated with red sanded bark extractive. So also in this case, you can see because mango rubber wood is have low density, and its anatomical structure facilitate the impregnation. However, mango wood and Melia dubia is some density is more in comparison to the rubber wood. So this, you can see the retention is lower. Again, Melia dubia wood, same procedure was carried out. And weight gain percentage, we have also calculated weight gain percentage. This is different extract, how they behave in the wood. So you can see in the graph that HB Hevia brasiliensis means rubber wood is the for treatment 1 means 60-minute experiment, And treatment 2 means say 120-minute experiment. So after the long duration of time, the extractive intake is something more. The same with the Milia dubia. Mangifera indica means mango wood, and MD means Milia dubia. So in second set of experiment, the intake was more. The similar is this is impregnation experiment continue. This you can again see the in set 1 the Hevia brasiliensis means rubber wood is more in comparison to the other two wood, but in the same 120-minute the intake was more. Now we have after the impregnation of the extractive into wood, we have studied the antifungal activated of impregnated sample as per the Indian standard 4873 for part 1. In this, we have the treated block was placed inside the fungus, this bottle having the fungal with the media, and this experiment is carried out for the standard says that the 12-week experiment is there. So 3 months, this experiment takes 3 months. So you can see here 3 months continuous it is kept with the fungus, and the loss was determined how the wood was fungus attacked and eaten the wood. So this treatment loss was determined after 3 months conditioning. So you can see the water extract loss was 4%. Second experiment, acetone extract, the means ki antifungal activity is more in this water extract having less. Acetone extract, this methanol extract is second, third, and methanol acetone extract was least degraded by the fungus. In comparison, this is comparative with the other plant extract. The similar study was carried out by the Akhato et al. Why I have kept here? To show that the red sander bark is more effective than the one study was carried out by using the pine needle extractive on bamboo blocks. So you can see here the loss was 13%, 9%, 4%. But in the case of red sander bark, the loss is so less in comparison to the pine extractive. So in this also you can see the red sander bark is more effective to against the fungus. The color study on the impregnated rubber wood we have carried out. Here L star means lightness or darkness. A star means redness and greenness. Positive A is red, negative A star is green. Similarly, B star for yellowness and blueness. Positive B is blue, yellow, and negative B is blue. You can see we have also carried out the color study of the impregnated wood. This is for treatment 1, this is for treatment 2. Here very interesting fact I want to show. This is accelerated anti-termite testing is also we have carried out, although this is not committed in the project, but to give a holistic approach, we are including this study because of termite also we should know whether it resists termite or not. So when rain will come, so after, because the termite study is more suitable in the after the rain, occurrence of rain. So we will carry out this experiment after 1 or 2 months when after rain. You can see here the coating experiment. We have also studied the coating of the different extract on the wood because impregnation require more infrastructure, high big unit, big pressure unit, big vacuum applying unit. But in the coating experiment, no that facility is required. Simple extracts and we can apply coating. And also in addition to that, coating requires less extractive and give more attractive color. I will show that. You can say delta E means total color change. It reflects the total color change of the treated wood. So this is I can show. the water extract, alkaline water extract, aqueous methanol extract, acetone extract, and methanol extract was applied on the rubber wood. And this is the exposed sample. This is the unexposed sample. Exposed sample, you can see slight change has happened. Degraded means ki after this is applied 250-hour continuous UV light was applied to see its resistance towards the weathering. So you can see after 250 hours of continuous exposure, very little change has occurred in water extract, in alkaline water extract. This is methanol extract, this is acetone extract, this is methanol extract. So this is so that it is very effective also. This is so the color change. You can see minimum color change in the water extract, whereas maximum color change is in this acetone extract. See, this is a close view. Coating experiment. This is control sample. This is rubber wood. This is alkaline water extract. This is water extract. This is methanol extract. Acetone. You can close. A is exposed portion. This is unexposed portion. Very interesting thing. This is wood. Piece of red sander wood. So here I want to show that this looks very close to this wood. Actually, the all wood 1, 2, 3, 4, 5, 6 is this wood. This is uncoated. This is coated with the different extracts. So you can see the wood is close to the real wood. Coated with the red sander bark is very close to the wood. Not only by looking by human eye, it can be seen with the numerical data of L star, A star, B star, where in the control L star is 79 and wood is 27. This is L star value of the wood. This is A star value means redness is in untreated wood is 5, whereas wood is 17.8. B star means yellowness in it is 22.95 because it is yellow, and in wood it is 12. So methanol extract find very close to that. 20, 25, 27, 17, 17, 9, 12. But we can also make after more concentration or more application, we can make anything, any appearance, because wood also vary. This wood is looking like that. Some other wood appears more darker. So I want to show that by applying different concentration and duration, we can make very close to red sander wood. So imagine this is make of rubber wood, suppose, and coated with the red sander bark extract. How the feeling would be? Oh, it is coated with the red sander bark. Why the wood is going to costly? It's also application finding in medicine, therapeutic activity. So we have thought that the bark should be used for some economical purpose. So we have carried out the coating also. We can see it. This is very encouraging. Now we can prepare this wood to the very close to any of the wood, or whether it is darker wood. I agree that this is wood of red sander may have some darker appearance, but we can, if we increase the concentration, that can be matched to the real wood. So these are the data showing that the different color change after the exposure of 250 hours. So variation in color change with increased in exposed duration. You can see this is color change in 24 hours. This is 50 hours. This is 100 hours. We have recorded control value in after 24 hours of exposure, 50 hours of exposure, 100 hours of exposure, and 250 hours of exposure. So you can see here the control is this one. This one is water extract is very little color change has come up. Whereas in acetone, some acetone is green one, see the color change is more. So methanol extract is also very, this aqueous methanol is very less color change has. So this shows that if we apply on the rubber wood or milliard wood or mango wood, the color change, the color degradation will be retained for longer period. Again, this I talked in earlier. So you can see here the red sander bark extractive and this is wood extractive have some common HPLC peak at retention time at 6.74, some common peak. So by this way, we can say some component may be similar. But why we have carried out this? To ensure that after any customer can find whether how can I be satisfied that this has been coated with the red sandalwood. So in that case, this is although initial study, we can take a very small piece, a small only we can apply on the lower side of 2-3 drop of methanol, and we can take this solution, inject on the HPLC. We can come with that will come closer to that this value. So we can have some idea that guarantee the consumer that okay, make sure of them that okay, it has the red sandal bark extract. Otherwise, what people say, oh, you are saying this is coated with the red sandal bark. We used to go to the market purchase. Even the seller, the retailer say that this is make of teak. But many of us can understand they can sell any other wood in the name of teak. So similarly, we have carried out for that purpose. This is HPLC of bark. This is HPLC of wood. I have shown the data. Another also we have carried out UV analysis of wood and bark. We have find some common peak, although not isolated and characterized the compounds that is taken care by Deepak ji and Srivastava ji. So they are. So this is you can show these similar peak are observed. So conclusion of till now, the impregnation, the dry wet retention of the extracted impregnated samples were found to be satisfactory with the maximum retention in the case of extract of water and least in the methanol extract. Moreover, a higher retention in rubber wood was followed by mango wood and then Milia dub. Suggests that the low dense wood has a higher retention compared with the high dense wood, and higher time duration during the pressure treatment resulted in higher retention of the extractive, which I have already shown. The antifungal activity, acetone extract was found to be most effective compared to the other extractive. Water extract showed the least efficacy. However, it is still provided comparable effectively when compared with the control sample. And extracts were found to be more effective against this Trematis hirusta and Oligoporus placentas, and higher treatment time provided better antifungal activity. I have shown that 2 hours has better large antifungal activity, more antifungal activity. The third one is anti-UV efficacy was higher total phenolic content. We have also calculated that is, and also it is going on. Water and alkaline water extract were found to be most effective in protecting wood against the UV radiation and thereby reducing phytoyellowing. However, methanol and acetone extract showed significant color change through, though this finding can be used to demonstrate requisite set of timber and use in appearance similar to that red sandalwood. So in conclusion, till now we can say this study shows a very good result for the purpose of coating and impregnation of bark extractive into the wood. So I thank National Biodiversity Authority for funding this project, and I thank Andhra Pradesh Forest Department for giving us the red sandal bark for the purpose of this result. And recently also I have discussed with Gopinath sir and sir with for the more red sandal bark we are requiring. Sheikh Salam Sahab I have already discussed with, he will be providing in a week, sir, they have told. So I am again expressing my thanks to all of you. Thank you, sir. Any question?
Thank you. Are there any questions?
Sir.
Does it add value to the life of the wood?
Sir, that is why, sir.
How much? What proportion?
The antifungal activity, because wood is degraded by the fungal attack and termite attack mostly, and weathering, if we sunlight UV weathering. So it is very good result in the terms of the weathering also I have shown, sir. antifungal activity, very good activity has come up. So we can say termite activity we will do after the rain. So you can, sir, see that it is in condition of durability. I agree that the appearance is good, but durability also, sir, if we apply or impregnate, durability will enhance. Sir, that in year wise, I can say I will be able to tell after some time, because this anti-termite activity will I do. But as far as weathering condition is concerned, sir, I can say in exterior this will retain for 4 or 5 years, and if internal means not exposed to outside, in house it will more duration. The color will not fade away, and fungal attack will not be happen. So you can see, sir, if we are using in house, inside the room, this will go more. Durability will be more.
All right.
Sir was
you.
asking something. Kurin Kanan, sir.
Yeah, it's already a lot of fake timbers are coming in the market. So you are providing some more idea for making it, for making the mango timber into a bringing as a red sander timber.
No, sir, this is not faking, but
No,
why?
Yeah.
Because if you will keep in one hand red sander, anyone can identify why
that is there.
it is not red sander. But appearance wise and feeling, this is aesthetic value, sir. Suppose this is coated with the
Yeah, that is correct.
red sander bark extract. You will feel better. Oh, this is coated with the red sander.
Yeah, actually, I am affected with that, because during my construction of my house, I took this one, Padak. They mentioned that it is a Padak, but within 9 years, all the thing is eaten by termite. Then we just take it out and just made a piece. The color actually outside is the Padak color, but inside it is white. So it is happening.
In the case of impregnation,
Yeah.
through and through color is there.
Yeah, that is there. So,
You
but
only-
coating will.
Coating. Impregnation through and through, for descent to descent.
So I am not criticizing your work, but this is an aspect of the.
Sir, this is a value addition of
It's okay.
less durable wood
Yeah,
and value addition of bark. Sir, sir can tell, Gopinath sir can tell, sir, there is no price for the bark. But if we can establish that this can also have economic value. Yeah, useful application. Implications. Sir, there will be, of course, sir, there will be cost, more cost will, if the only cost is the bark. If the bark will be available, this requires the how much price of the bark is. Otherwise, application and time and labor is same, sir. Right now it is not, sir, but if we can think over some useful application, the price, the forest department can get some price of the bark also.
C-grade quality. In Goron, sir, we have a lot of thousands of tons of C-grade quality, sir, and non-grade also. It's very breakable and fragile, no export quality. Can we make that powder and make into the restaurant wood, sir?
This is wood. You have a bark or wood?
Hardwood, sir. C-grade hardwood is there. We have. The technology we can use here.
Inferior wood. Okay, sir, it is more, if we take out wood, would have more extractive, 17, 18 or 20%. And wood, then it is more, the 5 times better than the bark.
We can get again residence wood out of it, quality wood.
Sir, if we can provide that is inferior wood, the question is the getting the wood. If I talk the red sander, the forest department personnel are so attentive on red sander, they can listen who is talking red sander. So if you, sir, my request is you please provide some waste, what you are talking, the waste material or less inferior quality of wood, we will happy to do that, sir. That will be great value addition.
All right. Thank you. Thank you. Maybe we can move on to the last speaker,
Thank you, sir.
Dr. Prabuddha. Can you try to?
Good afternoon, respected Chairman Sir, Secretary Sir, Senior Forest Officers, scientists, and my colleagues. This is the last presentation, and probably I am the odd man out among all the accomplished scientific community. I am a Indian Forest Service officer, and I am presently working in Institute of Wood Science and Technology, and it is a collaborative project between Institute of Wood Science and Technology and the Transdisciplinary University. Being a forest officer, I more concentrate on the field level observations, and my colleague Dr. Mohan is more concentrated on lab oriented work. So our project is about development of SSR markers, simple sequence repeat markers, and assessment of genetic diversity of natural population of red sanders distributed in eastern Ghats of India. And we know that molecular markers are very useful in assessing the genetic diversity. But there are reviews more on use of the random markers like RAPDs and ISSRs. And SSRs, or simple sequence repeats or microsatellites, are more robust markers because they are codominant and locus specific, and it is having high repeatability. So we took a project on developing SSR markers. So I skip all these initial slides. Coming to the objectives, the first objective is to develop simple sequence repeats, Pterocarpus santalensis, using the next generation sequencing. The only drawback of SSR markers is its development. Even though they are very robust, its development using the traditional method is very tedious. But with the advent of the next generation sequencing, which gives high throughput, it is more easy to develop SSR markers. So we have adopted this technology for developing SSR markers. Then acquisition of germplasm from different natural population and to identify the polymorphic SSR markers. So we consulted with senior officers and the field level officers, and apart from the literature survey, and we could cover all the major natural red sander growing areas for our sample survey. And the third objective is to assess the genetic diversity and population structure of natural population of red sanders and identify the population specific SSR markers. This is, if possible, the third one is. Because population specific SSR markers is a very unique thing. So this is our work plan. I'm going to skip it. So these are all the sample collection areas. From the northern end at the Nandyal division, Rudravaram, we collected all possible areas, and we could collect more than 372 samples from 22 ranges and 8 divisions. So these 8 divisions are in 3 circles. So while sampling, we have collected the samples from seedlings, that is 0 to 10 girth class, and 10 to 20 centimeter girth class, 20 to 30 and more than 30, that is mature seeds. We wanted to capture the genetic diversity from seedlings to mature crop. So these are all the places we visited for collecting the samples. And we used modified DNA isolation protocol for isolating the DNA from red sanders. Red sanders being having high phenolic content, it is difficult for isolation of DNA, but we could succeed by modifying the method. And it appears to be very similar to the IFGTB studies, but we sat together and we restricted our study for whole genome sequencing. And they have done it on transcriptome studies as they have discussed earlier. And now I hand it over to Dr. Mohan, who is more working on the lab-oriented research on genotyping. Mr. Mohan.
Thank you, Prabodh. So we initiated with, when we got this project, there was a COVID that time. So we have screened all the papers published in the area of especially Pterocarpus centellinus. And we collected all the published literature, as well as there was a one Pterocarpus erinus, there was a one sequence was available. And based on that, so we have I identified some of the SSR markers from the published and as well as from the genome sequence of the erinus. And then we could able to identify the 4 from erinus and another from the paper from 2 polymorphic, 6 polymorphic marker from the already published literatures. And these are some of the gel photos. And so we have and the second approach was, so this was actually a proposed project, whole genome sequencing and identifying a SSR marker and to study the genetic diversity analysis. So what we have done is, so we have isolated a DNA and using a Illumina paired end sequencing and as well as nanopore sequencing. So nanopore sequencing is still, we have to get a result, but Illumina sequencing we have got, and we have assembled with 2 or 3 different types of assemblies and then identified SSRs and designed primers with the M13 tailed and for genotyping and especially fragment analyzation, and PCR amplified product was master mixed and then fragment analysis has been done. And this is the coverage we got, more than 100x coverage we got, and these are the reads we got, and these reads has been converted into quantics, and using these quantics, so we have identified the SSRs, the different types of SSRs in especially Pterocarpus. In the mono repeat, A is highest and followed by AT and AG and AAT. So we based on this data, so we have designed, selected, and also we have done an assembly of 3-4 different softwares, tools available in the public domain. So more or less, so all of them are gave us similar results of SSR distribution in the Pterocarpus centellinus. And we designed 35 different primers, and we have M13 tailed. So these are the primers, and we have standardized the PCR method for primers. And one of the major reasons, so there is a polyphenolics which actually inhibit the PCR, and then we have overcome with that also now. So everything is working fine now. So from the designed primers, we have got 11 polymorphic markers for utilizing the population screen. And this is the preliminary data what we have, whatever the analysis so far we have done a statistical analysis of that. Especially Pterocarpus follows very low observed heterozygosity in the natural population compared to less than the expected heterozygosity. And also it has follows the moderate genetic differentiation, and this could be only, it is just we have taken some 15 to 8 to 12 populations. So now we have to get a holistic view of results from all the populations. So what we observed is, so we have got the total number of alleles around 203 from the so far what we have done, and so it ranges from 2 to 43. And polymorphic information content was 0.3 to 0.95. So there are something more than 11 loci as giving more than 0.9 as a polymorphic information content. So in the preliminary, whatever we have done, so it is showing very low observed heterozygosity compared to the expected. And also observed alleles was 5, and then the effective allele number is 3.9, and inbreeding coefficient is moderate, and also it is indicating so that is a moderate level of differentiation between the population, and there is a high gene flow currently whatever the population we have used now. So and for 4 primers we are able to, 4 loci we are able to complete for all these populations which we are collected. and shows again here, so there is a low level of observed heterozygosity compared to expected, and also the moderate level of inbreeding coefficient as well as genetic differentiation. So next level, so what we thought is these are the collected different places what Prabhuda was mentioning. And then so we have done an ecological niche modeling where they exactly belong. So whether they are high suitable or low suitable or moderately suitable and all. So these are the 22 and we have classified based on the niche modeling. So there are few of the populations are very high and some of them are moderate and some of them are excellent. And then we want to fit it all these studies into this group and see where their genetic diversity is very high and allele richness is very high, so that we can take a conservation measure and as well as propagation as also as well as we can display as something which is like a hotspot of genetic hotspot, so that we can future regeneration can be done with those populations. So overall summary, so we have got 372 leaf samples from 22 populations, and we have done an re-extraction as well as PCR failed, everything we have sorted out now, and cross amplification led into 6 polymorphic markers, and whole genome and de novo assembly has been done, and then we have designed a primer, and we have identified SSRs and designed primers using tools, and 33 primers were designed, and out of 33 there are 11 are polymorphic markers. And so genetic diversity analysis done for 8 populations with the four polymorphic markers. So it reveals actually low level of heterozygosity compared to expected heterozygosity level, and level of genetic differentiation among the population. And there is a low level of current result shows low level of inbreeding coefficient and high gene flow between the populations. And but this is a preliminary study, but we have to complete all the population and get a holistic view of that analysis. So this is the future plan of work. So developing hybrid assembly and then getting some more SSRs and amplifying with all the 24 population and do the statistical genetic diversity analysis, and also fit into genetic analysis across niche areas of the 4 or 5 different groups of niche distribution, and finalizing report and publication. So we acknowledge so NBA for funding, and as well as Andhra Pradesh Forest Department and all the officers who are helped in our collection, and also working team both in IWST and FRLST TDU. Thank you.
Thank you.
Overlap between the work presented by you and that of sub-project number 3 of Dr. Kunikanan. So how do we resolve this?
No, what I understand, they have used an EST SSR marker and then only 6 populations, and those populations we have not used. It's
So can
completely-
at some time we can assimilate
Yeah.
both the findings and come up with the
Yeah,
common.
yes, sir.
So that one thing is there should not be variation between your reports.
Yeah,
And
I
it
don't
should
think there
actually
will
add
be-
to value.
Yeah, it will
Whatever
definitely
it
it will
is.
add a value. Both those are all genic markers. What we are
Another
using
thing
is-
I wanted to ask you.
Yes.
Was maybe I can talk to you a little later. Just go back to the previous slide. Previous one. Previous one, yeah. No. Yeah, somewhere you have mentioned the low inbreeding,
Yes, sir.
low inbreeding and the low
Genetic
heterozygosity.
differentiation. Low heterozygosity, sir.
Low heterozygosity means they're more homogeneous
More homogeneous. Yes.
and but low inbreeding.
Low inbreeding because they are very small packets. So there is a gene flow is happening, but still there is a. Sorry, sir. Yeah, that's what. So that is what. So there is a moderate level of gene flow is there, as well as differentiation. It is not high differentiation.
So they're almost pure crops.
Yeah, it's not completely pure crop, sir.
Almost, I said
But
almost
it is-
pure crops.
Again, I cannot conclude because this is only 8 to 12 population we have
And
done.
you also said some 5 alleles and what actually, can you elaborate more on that?
5 alleles, low size, sir.
Somewhere you mentioned it.
This one.
Yeah, what do you mean by 5 alleles or 6 alleles, whatever it is?
This is the allele, sir.
Ah,
So each loci has a given 4 different alleles.
okay.
Okay. And alleles varies from 2 to 43. So there is a one of the allele which is very high, which is somewhere 43. This allele, this loci of marker.
I'll
which is
talk to you
giving-
in person
Yeah,
afterwards.
yeah.
One last question maybe from the back, yeah.
Hi, very nice talk.
Mic's not on.
Hello.
Yeah.
Hi, very nice talk. I had one thought. Genetic diversity, this low heterozygosity, could it be because of fertilization between male and female flowers on the same tree?
Yeah, that is, it is not.
But the bee could visit the male flower first and then the female flower on the same tree, right? So then you might expect. Okay, I see. Also, geographic level differences in populations. At what distance scale did you recover these patterns?
No, we have not that still derived at a geographical scale. Okay, this is what we have done is, so there is a one patch. So 8 population we have done, they are geographically they are very close. In aerial distance is very low. So that's what we have done in so ecological niche modeling and see where actually geographically how they are different and is there any barrier between one to there. Okay, so that analysis has to be done. Geographical distribution and versus genetic diversity, allele diversity, everything we can do, but it is under just preliminary results. These are.
All right, thank you. Thank you. That concludes our session on red sanders. And now I believe that we divide into 2 groups or. Okay. Okay.
And what I would like to request our business members that we can shift this review meeting after that. That means you can continue with the same program now.
Okay, okay. All right.
Everybody attends that. And once we complete that meeting,
Sure. So we move ahead then. Sure. So would you like us to finish tea before your meeting? No, before your meeting, would you like tea to be finished before your meeting or? No problem. Okay. All right. All right. So can we move to the first research talk? Mr. Ashok Kumar from WWF. Again, these are short research talks, so 15 minutes with 12-minute talks and 3-minute discussions. Okay, if we can try to stick to time, we can catch up by the end of the day.
So good afternoon, everyone. Myself, Ashok Kumar. I am a senior project officer from WWF India, works in Hyderabad State Office. So today I'll be presenting on the previous works and the present work we are involved, WWF India is involved on the biodiversity conservation in the Eastern Ghats landscape. So WWF India focus areas are both Andhra and Telangana. We work on the biodiversity conservation. We also work on different taxas and we also work with local communities, primitive tribal communities called Chinchus. Like sir already informed in the morning, Mathu sir, that any conservation we have to do, we have to also take care of the local communities reside near the protected areas. We are also involved in the policy and advocacies, like we involved in the as a advisory committee members, and we also work with the citizen network. So we do nature trials, eco trials, and we also conduct a EarthR program in the March 26, where switching off the lights. If you can see, we try to convince the governor's bungalow and switch off the lights. So these are one of the citizen science work we do. And we also conduct the amateur naturalist training programs, where once the training is conducted, we will be using the volunteers for the trainings and census programs which conducted by Forest Department. So the first study by WWF India in Eastern Ghats landscape was in 2004 and 5. So it's a remote sensing and GIS based study, which started from mid of the Eastern Ghats in Chittoor and till Tamil Nadu. So we conducted this to identify the degree of fragmentation of the patch size and the forest, and found that forest cover analysis shows that 38% of the study area is characterized by forest habitat. So remaining everything is with developmental activities as well as anthropogenic disturbances. And several floral elements of this landscape are narrow distributional range, and any adverse impact can have a serious impact on the floral content. Like Sir said, Playas said in the morning, like very few species you can see in a few landscape, few micro habitat type. If you lost because of the developmental activities or any of the anthropogenic pressure, then we may have a very tough time in the future. So we also identified in the report the priority areas where identified endemism and threatened species also, and for immediate and interventions. And we also did the biodiversity survey conducted for intensive showcase of the importance of Eastern Ghats. Total 10 publications were published from WWF India from 2009 till date. It's on all the taxas. And we also have an intensive study for the 2 pictorial field guides we published. One is biodiversity of Nallamala Hills, another one is biodiversity of Araku Valley. So the outcome of the study is published as a book for the tourism department and the forest department to promote ecotourism. And we found that total number of 40 floral and 134 faunal species described in the Nallamala Hills biodiversity book, and total 52 floral and 122 faunal species described in Araku book. And this Eloganthus radiatus, a copper-headed trinket snake, we found it in Araku. And this Buthoscorpia royalensis, which is on the Rayalaseema thing we described it, it is found in the Nandyal in Kurnool district. So presently we are working on the tiger conservation in Eastern Ghats, both AP and Telangana, but I'll be focusing mainly on AP. So we work on this landscape, starting from Amrabad Tiger Reserve till Sri Venkateshwara National Park. The importance of this landscape is like you have 2 tiger reserves, and almost it has 3 wildlife sanctuaries and 1 biosphere reserve. And sadness of this is it cuts several national highways and state highways, and almost 42. This you can see this area, this part. We have around 42 small villages and townships, which makes this landscape very crucial. So it is about the size of 13,628 square kilometer. Next slide, next slide. So conserving large carnivore is a very big challenge because we compete for food, we compete them with space, and livestock depredation is there, and carnivore kills even vehicle carnivore sometimes. And it has a long home range, it's solitary and very low in density, so sighting them is very hard. So keeping these challenges into concern, we prepared a project which we are working on. The goal is to conserve the tigers, prey, and their habitat in the Eastern Ghats landscape, as well as to make the connectivity working. So main thing we are doing is this, like if you see the management effectiveness evaluation report of 2018, the main thing what they mentioned in the weakness of the tiger reserve is that capacity, motivation, lack of motivation, and the capacity building of the frontline staffs in the wildlife management. So we take this as serious concern. We try to build the capacity of staff to enhance the management effectiveness, especially in the tiger reserves and the other protected areas, which I mentioned from Nagarjuna Sagar Srisalam Tiger Reserve till Sri Venkateshwara National Park. So we conducted training need assessment surveys. This assessment will be like on 3 phases. One is assessment of the previous surveys conducted. Like whatever the trainings already they have undergone, we will be evaluating it. And we'll be seeing where the gap is, and we will be trying to fill the gaps with giving training programs. And second one is the staff perception surveys, where we will be interacting with the staff as well as the local community over there and bringing a bridge to both to have a successful conservation. And next one is the support need for the frontline staff as well as the base camp. So during this survey, what we do is we have a 3 different training program for 3 different layers. One for the production watchers, next for the beat officer and range officers, then with the DFOs. So we'll be knowing what exactly they need. And later we go to the base camps, critical base camps in that this one corridor area, and we will see how the status of the base camp is. Then what we can provide for a personal front as well as for the base camp to get a better protection. So as part of the capacity building, you all are aware that All India Tiger Estimation is on the 5th cycle we are going on. So presently the camera trapping and all the steps are going in all the tiger reserves. And for that, this is the framework. So basically what they do, people who don't know about the background of All India Tiger Estimation, they collect this is the phase 1 data which is collected from the forest department for all the beats in overall India. For 8 days they collect. All over forest division in India, they collect the data and they collect the indirect signs. They collect the data on the prey, indirect sign, vegetation, human disturbance, and the dung count. This has been collected with the help of a digital application called Impregnation software. So once this is done, the data will go to WIA, where they will use this covariates on remote sensing and GIS to bring a base for camera trapping exercise. So once this tool is ready, we'll go and deploy cameras in the forested areas where already a camera trap has been applied. For example, if 2021 the camera trapping is going to be done, so already 2018, wherever we fix the cameras, we'll fix there only, so that to get the change in the trend and status. So these are some of the glimpse of the training programs we conducted. So we conduct training to the forest department on how to use the cameras, how to use the mobile application. So this is a mobile application I was saying about. It is called M-Stripes program, where you can collect the data with the application. It is a geotagged one. Each and everything of yours can be recorded and geotagged, and you cannot manipulate in it. So it's not a business of like somebody sitting in somewhere and writing, I have seen this and it's, and you have a pictorial information in this. So everything is recorded. Your track will be there. So total 573 staff from 22 divisions, including the computer operators, were trained for this mega exercise. So next we are working on enhancing the use of key corridors. The corridor which I was speaking on other wildlife connecting NSTR and the Venkateshwara National Park. So we are working on assessing the status of tigers as well as the prey population through the forest department camera trap records. So we collect the camera trap records once the camera trapping has been done. We collect the camera trap data from the forest department and we identify tigers and we identify leopards with softwares. And what about other species which doesn't have marks? Tiger and leopard have marks, but what about cheetal, sambar, or wild pig and all? So we come up with the abundance index. Okay, the places like what do you say, NSTR, which is a larger in size, and the detection of ungulates is very rare. So if you go for 15 times or 20 times, you walk a transect, you can see 4 or 5 cheetals, hardly. The people who worked in that landscape will know better. So what you can do that in case of that, we use abundance with the help of camera trap data. And if you see, this is a corridor which I was speaking. It's a 2011 night lights data. Okay, you can see the human habitation. It is in 2011, and now you can think how big it would have been developed. And you can ask like, how come we come with this data set? How come? You can see the information over there in the camera trap images where you have a date and time. So what we do to assess the threat, we segregate the camera trap data like tiger separately, leopard separately, livestock separately, dog separately, vehicle separately, human separately. Then with the help of the time activity data, we prepare, for example, this is one of our work where you can see that the gray one is a cheetal one, And the dotted line is a human disturbance. So wherever the human disturbance is high, you can see that cheetal activity is low. So this kind of threat assessment we can do with the help of the camera trap data. And so this is the camera trapping data which I was speaking about. So the cameras will be fixed on a trials where 2 cameras will be there. So if one misses, another one, and both the flanks are important for us. In case if only one flank we are getting either left or right, then we can compare with other things. So we fix 2 cameras to get both the flanks. And we get the images like this, tiger images. So what we do, we get the tiger images from here, and we have a special software called Extract Compare, where you can identify the tiger individual. See, you can see this tiger and this tiger are same. So if you have like 5 or 10 images, you can do it. But people have like 1,000 images of tigers or hundreds of images of tiger. You depend on software to do this. And this software works on the logic method. So you have the dots here where it constricts and puts a flank, and it gives you the individual thing. This also helps for the population estimation as well as to detect the poaching. So WIA has a national repository of thousands of images of tigers of all over India, and not only India, the Bangladesh also, Bangladesh, Nepal, like that. So if any tiger in somewhere in Srisailam or somewhere in Tirupati, it has been poached, and if we can get the skin photograph, then we can compare with the repository and get the information where the last tiger has been sighted. So these are the few things we are working on. And we also work with the local Chenchu communities. As I said, these are primitive tribal groups. We provide them the sustainable livelihood options in the tiger reserves. Like we conduct a baseline service wherever they stay. It's mostly in the tiger reserve, but we also want to do in the corridors also. And encourage we mainly want to encourage the Chenchu women over there, especially the women, by training them in providing alternative livelihood to reduce the dependency of forest resources. So we train them in making the paper bag and leaf plate making. And we also train the local Chenchu tribal youths for as a nature guide. So this Srisailam Tiger Reserve as well as the Amrabad Tiger Reserve, all these are like tourism area where the people come for the pilgrims. So we help them in getting a nature guide so that they will financially they will be stable and they will not much depend on the forest resources. So, and next one is this is our pioneer work. We started in 2011. So this is a water management. It's a solar-based deep well pumping. So what you do, we have first installed in Nagarjuna Sagar Srisailam Tiger Reserve, and now it is replicated in many tiger reserves as well as other states also. But it started in NSTR only for the first time. So it's mainly a solar driven. You can automate the timing, how many hours in a day you want to, pump the solar water. And it works like from 10,000 liters to 30,000 liters water you can withdraw. Especially during the summer, it is very much helpful where you can see a water body in air. The water goes there and the animal, like you can use for the summers, especially the animal takes it very carefully. Until date, we have 30 pumps have been installed in NSTR, and it has reduced the human wildlife conflict very much. It's beginning to narrow down. There was not much conflict, especially these places which are dry deciduous, they get conflict with the sloth bears and this one, wild boars. Now it has been drastically reduced after this pumps. Like I already said that we also work with the policy and advocacy. So WWF India lends its knowledge and advisory services to the following committees: the State Advisory Board for Wildlife and Andhra Pradesh, and for BioSAP, Kempa. We are also a tree protection committee member, and for ZAPAT also we are. And the importance of study here, I just go back once again here. So according to the report, it says that this landscape, starting from Amrabad till Sri Venkatesh, can sustain 400 tigers. It can sustain 400 tigers. Presently, we have 8 in Amrabad Tiger Reserve, and in NSTR it is 38. In Sheshachalam it is 8. That was recorded tiger individuals in 2018. And for our surprise, out of that 8 individuals in this one, out of the 8 individuals, which is camera trapped in this area, there are only 3 captured from this Prodatour division of Kadapa and Prodatour division. Remaining 4, the tigers are moving from here. From the top, from the GBM, it comes here and it goes back. Okay. So it comes from here and there is a movement of that 4 tiger individuals and we will also increase. So you know how important to connect that corridors in this. It's very essential to connect the corridors. Like I said, there are 42 villages which was identified, so small towns in 2011 by NTCA and WIA, which is very crucial, which acts as a barrier restriction for the movement of the animals. If we have any alternative plan to mitigate this, because the tiger population in Srisailam, it's increasing. Once it is increased, it will come actually downstairs for survival. So if there is any problem in this one, if we cannot manage it, then the local population will have a bad fate in the future. So, next one. That's it. If any questions, thank you.
Does anybody have any questions? All right. If not, we'll move to our last plenary speaker of the day. Before we do that, I'd like to welcome Professor Satyanarayana. He's the director of IIT Tirupati. He just joined us. Welcome to the meeting. Maybe we'll have him say a few words right after tea. After this plenary, we'll have a break. So our next plenary speaker is Professor N. Parthasarathy. Professor Parthasarathy is from the Department of Ecology and Environmental Sciences at Pondicherry University. He's a forest ecologist and has conducted extensive research on tropical dry evergreen forests in many of the hill regions of the Eastern Ghats. He's one of the few ecologists in India doing long-term research and has documented forest stands for around 3 decades at some of his sites. His areas of expertise span across terrestrial biomes, forest botany, medicinal plant resources, functional ecology, and the dynamics of tropical forests. Dr. Parthasarathy, thank you for this.
Esteemed colleagues, scholars, and students, it's indeed a pleasure to be here at IISER Tirupati, briefing our efforts over the last few decades, parallelly in Western Ghats, Eastern Ghats, and Coromandel Coast forests. Here I'm just briefing the baseline inventories of ecological studies in the Eastern Ghats and the way forward for IISER Tirupati. Most of the products are our own first generation and the next generation student communities here from Pondicherry University, be it here or IISc or A3. So that gives us a pleasure. There is a continuity of ecological research over a couple of decades. And I have a couple of suggestions before IISER Tirupati Research Unit. And particularly, I make this mention in this platform under the NAP. You'll have multidisciplinary research facilitated through various means and small projects. So with this briefing, I shall move on to the topic. Well, we have heard about the Eastern Ghats, the extent and the other details of 6 states it's covering, and I skip on those basic details. And it has been a great opportunity, various scholars conducting research, and I'm just taking the sample research that we have had in the Southern Eastern Ghats region, covering 6 hill complexes, and over a period of about 15 years, we could have data generated on the ecological standing of the plant life forms in the Eastern Ghats forests, and many of these could now go to the global tree database and the global liana or the woody climber database too. So inventories, people do take up from the smaller to medium scale to larger spatial scale that would have the advantage of addressing various aspects of ecological research, and when monitored on a long-term basis has great advantage with respect to what is happening to the system and the extent of resource base that we have in the sample forest area. So that way, the global long-term ecological research programs spanning across the world, taking into account a hectare to as large as 52 hectare areas in many of the CTFS funded programs, forest geo programs. And standard protocol is being followed in many of the studies, but this is against to state with the ICER Tirupati that there is no compulsion on sticking on to under the name of standard protocol set in by CTFS alone like, and that was a lesson we could learn over the last 3 decades funded by 6 different Government of India funding agencies, that you should tune to your requirement and the nature of the study under the given academic framework, but essentially without compromising the core or focal research programs in mind. And it's better that some minimums are always met with, because comparing the comparable being a logic in science, it would help our study to what extent stands with respect to the scientific data generated over years from many other geographical locations. And standard methodology with respect to data synthesis and analysis. And all these will eventually help these institutions to be mapped on the regional and the global scale. The valuable scientific contributions that will go on for global synthesis. That was a simple effort ever since the first batch of MSc students took up, and those have first initial 6 years, nothing could go because 1996 only first computer to the university. Data storage started from then, but what has gone into is from 1992 onward, somehow with the hard copy that we had, all those data now could go to Global Tree Diversity Database and the LINA database too. So these would have the advantage of addressing many ecological questions, not only the number of trees, the number of species, and how are they distributed and how might the distribution affect forest folivores, frugivores, nectarivores, etc. And what are in particular the interconnections of floral faunal interactions. I shall be drawing little later your attention with respect to such interactions under the way forward lecture. So depending on the specialist pollinators, dispersers, etc., and all these studies, if it is a permanent research plot with respect to climate change and other environmental drivers, and this would go a long way to generate valuable data. So that way, emerging institutions like ICERs could contribute a lot over a period of time. The baseline research that has given us an idea of what is the extent of various plant life form diversity in various forests of the southern Eastern Ghats region, the large scale 60 hectare all India coordinated DBT funded program. 6 institutions, 6 blocks of Eastern Ghats forests, including SK University, Professor Pillaya's student, Dr. Ravi Prasad Rao, and the southernmost region for Pondicherry University, gave us the opportunity to explore at the larger spatial scale tree diversity, lion diversity, and the resource value of those species under the various ecological setting with respect to impacts, bioinvasions and human impacts, mining, plantations, roads, dams, etc. And these have give an idea about the extensive diversity we have and also the operative drivers. Well, the other important point with respect to scholars and researchers is to have their data and analyzed it thoroughly and encouraging them to publish, then only the scientific community gets to know about. This is phase one of it. Then the next comes to the mind that what is the resource base? The abundance data will be of great value, and it's a question of the long list of rare species that contributes to total biodiversity in a given geographical area and besides the dominant species. So such databases would help for next follow-up studies. I skip the pictures or images of many of these species, which are valuable in their own way. So way back in the early 90s, when we initiated this program in the Collie Hills of Eastern Ghats, and that is the time in the early 90s, large scale ecology was picking up ever since Campbell's book from Grinnell came out on titled Large Scale Ecology, and we have been on the contemporary mode of generating data at a fairly large scale. And what happens to that over a period of time by setting up permanent research, ecological research plots, the so-called LTERs in the 4 shola forests of the Eastern Ghats of India, and that has gone to the publication in Biodiversity and Conservation for one of the PhD scholars program. That is monitored by our MSc student after 3 years, getting back to the same site to detect forest growth, recruitment and mortality and carbon stock changes. Bharat is now with Nalanda University, and these are the advantage of permanent research plots. And one of the suggestions to ICERs will be that in couple of years, data generated that will give valuable information on what happens with the time to the system. So the Sherwarian Hills ever since initiated work in 1992 for the other PhD scholars program, and that has gone to again Biodiversity and Conservation for publication. These were some of the baseline studies from the 90s that we could initiate and later on extended to the entire southern Eastern Ghats. There are pockets of Eastern Ghats in the Kalrayan Hills imitating the Western Ghats in terms of their floristic diversity, and these sites are in danger because of the extent of anthropogenic pressure that are operating. And also between the Eastern and Western Ghats, up Dindigul town, we have the Sirumalai Hills, which is listed as Eastern Ghats sector, and there are a lot of pressures with respect to such sites, and these are some of the live examples of how sites are being deteriorating. And when we were with MSc batch of students, and there have been teams of students taking up research different aspects. And this is from the Kalrayan Hills of the Eastern Ghats on the structural aspects of the forests. The large-scale inventory of the 60-hectare plot formed one of the major breakthroughs in the All India Coordinated DBT funded projects. and for us, the southern Eastern Ghats region, and from Odisha, Dr. P.C. Panda's work to southern Eastern Ghats work through SKU works from Dr. Palaya's generations of research students. So that was published in Flora, and these gave us an idea at the larger spatial scale when inventories are conducted. Still, we get to have long list of species in low abundance, as low as just 1 or 2 individuals, and they form the bulk of the biodiversity. This is at the landscape level approach. I don't go to the details of it, but in the 60-hectare scale, 272 species and 27,000 individuals of tree species of 30 centimeter and above girth class, and how the species area curve, the estimator above and the actual realistic below. And in fortunately, couple of cases, it has neared asymptote, but not so in many others, attesting the fact that there are still more species one could expect, unlike the species area curves that we see in the T.C. Whitmore's book, ever increasing. At the fairly larger scale, they tend to stabilize. The inner details of the work. Then, when I compared the 76 years of tree diversity at the comparable scale of just 1 hectare, where does Eastern Ghats stand with respect to a hectare of plant diversity? At the hectare of 1 hectare scale, the biodiversity assessment. No, I'm not moving correctly. Back to the. Can you go back? No, you are moving forward. Back. At the 1 hectare scale, Central and South American forests, 300 species, most of the Amazon-centered forests. Back. Let me try, maybe. This is forward, this is backward. Am I correct? So at the 1 hectare scale, most of the tropical American, 1 hectare only tree diversity, adult species, 30 centimeter and above girth class. South and Southeast Asian forests, about 200. Northeast Indian Andamans, 100 species of trees per hectare. Western Ghats about 30 to 80, but 85 is maximum. Eastern Ghats about half of the Western Ghats, and in East Coast forest, tropical dry evergreen forest about 20 to 30 species, average being about 25 or so. When diversity is moderately high in one life form, proportional diversity we could expect in other life forms too, to make use of the resources provided by the basic structural framework providing plant community. So this needs to be kept in mind when diversity studies are planned, and this gives us an idea what is the extent of diversity across global forests at the comparable 1 hectare scale, and where does Eastern Ghats stand. There is an opportunity for the global tree researchers for the first time to pool the data for the cause of global synthesis, and that was from 2011 to 8/15, and by 15th June, we have the estimate of the whole of the tropics, 40 to 53,000 species. Then from the next 4 years time frame, last month the publication, entire globe, what is the number of tree species? 73,000. The global tree diversity contributors over several decades of the data generated. The next third paper in PNAS published last month on the extent of tree diversity. As of now, the estimate is 73,000 species. That way, quality work never gets unattended. So that is the quality works always will have their own standing. This gives us an idea what is the nature or extent of a level of endemism, how much are confined to the continental level. This gives the same will be applicable to the various sectors of the Eastern Ghats or Western Ghats too. The importance of regional level of biodiversity and globally just 0.1% common, and at the continental scale, the extent of endemism. That being the very nature of the pattern of global biodiversity, that gives us an idea of nature of partitioning across the continents and how much of importance needs to be given in terms of conserving regional level diversity. That's one part of it. The long-term plots have great advantage, be it forest geo efforts, the LTEO of Indian efforts, and various programs over the 3 decades time frame with our department. The core results were compiled in 2015 and analyzed that commonly gave the lesson that rates of tree mortality increasing in world's forests due to various environmental drivers. CO2 level increased by 16% in BCA Panama plot, 0.6 degrees Celsius increase in temperature, 30% change in the precipitation level, and landscape around the forest experienced deforestation, as it has been happening in many locations of the globe. For the last 2 decades or more, large trees all over the world have been dying, reported ever since from 2012. And at the same time, nature took care of in increasing the abundance of the woody climbers or the lianas, almost at the same proportion as a natural compensation to CO2 level increase. As you see, liana-clothed tree canopies in the East Coast forests. And Indian forests are the 3rd largest in terms of lion abundance, next only to the first Bolivian forest, 2nd African forest, nearing 3rd is the Malaysian forest, and India is the 4th largest in terms of liana abundance. And as of now, we have 4 source books on lianas ever since Charles Darwin's first book, then Putz and Mooney's Biology of Vines. Then we have 4 chapters in the book Ecology of Lianas, following our lianologists meet in University of California for a week in this 32-chaptered book. And then our own book with 13 chapters. These 4 books give us the state of art of liana biodiversity ecology and the importance of conservation under the context of climate change. When trees are dying, how much of compensation of lianas can make when trees are dying. The largest scale liana inventory from the southern Eastern Ghats region, 175 species, of which 143 species are with the resource values in terms of ecological or economic potential. The extent of leaf foliage lianas are capable of rewarding to the forest folivores and the fruit crop to the frugivores. Like you see Diploclisia glassescens in the Collie Hills of the Eastern Ghats, how much of vertebrate faunal communities are supported. Nothing is a waste in forest, including the invasive species, because the resources as ecologically or economically utilized by various communities. But the check is in terms of invasion, how much they are taxing on the natural biodiversity. So the climbing plants, various species of them, and lantana has been an obnoxious plant in various forests of the Eastern Ghats, taxing on the native biodiversity. And for the genus Jasminum too, one of the important bioregions. And this is how our students learn in the field in the forests of the Eastern Ghats. And they have exposure to the Western Ghats in the initial few batches, then Eastern Ghats, now on the Coromandel coast forests. So how much resource potential? In a phased manner, only such studies can be conducted by teams of people. And this is one effort on 175 liana species, how much they are distributed, abundance patterns of them, how much are resourceful to forest folivores and frugivores, and of economically valuable species. Just half a centimeter diameter liana, look at the extent of leaf canopy they can produce, a dioecious liana, male and female. So Pyrenacantha volubilis, and the fruits UAS Bangalore plant physiology team and genetics team. Anti-cancer drug camptothecin will be ready soon. They are in the final stage. Where this species is distributed from our publications, they collected the species and have been working on this for the last 5 years. Pyrenacantha volubilis. Understory diversity is equally high, but there are not many compiled works on these aspects. This is one of the gaps we have on trees, we have on lianas, but not much on the understory species compilation, because they are one of the sensitive components that could be kept as important candidates under climate change scenario. 1/3 of the world's landscape is modified for various purposes. Massive degradation is progressive. We need resources. Various biota in the wild need resources. We obtain from all the biomes. And at the same time, we are making use of the forest lands for growing various plantation crops of coffee, tea, pepper, and various other purposes, including plantations in various forests of the Eastern Ghats and mining for bauxite mining in the Eastern Ghats area. Under such scenario, where does we stand? This is an example of a paper in environmental science and policy with respect to the changes that are being happening due to various developmental activities in the core forest areas. And that was a concern we expressed in our first paper in Nature. And the baseline data has given us an idea over the last 3 decades in terms of what is the extent of biodiversity we have with respect to plant groups. And now, a couple of suggestions based on these baseline studies to growing institutions like ICER, what I have been incubating in mind, and we could come up with that in the tropical dry evergreen forest of on the Eastern Ghats, bringing out the book of our 25 years of work on the east coast of India. But when it comes to the patchy Eastern Ghats, the compilation can be made on par with, like we have Introduction to Tropical Rainforests by T.C. Whitmore. So we have been referring for ecological studies in many of the Western world books. And that has been the practice for many years. IISER, here E is there or is there. So, and under NEP, there are a lot of possibilities. These are few suggestions that came to my mind from the baseline research. Now it's time to have a fairly long-term programs of initially 3 to 10 years, ideally 20 years. By the time Nandini Rabin et al. Retire. So those compilations can come attesting the ecological research over a long term conducted. So about a 3 to 10 year plan can be kept in mind. Have in couple of in each forest type representative fairly landscape level large scale study plots for adult tree species, and for regeneration study you can cut down the threshold of trees and lianas. Number one. And address issues like biodiversity, initial inventory, it may planning for 1 year, and then execution for next 3 to 5 or 6 year program. Number one. Number two, it can be for various angles of ecological studies, biodiversity changes and the carbon sequestration potential in relation to climate change and other drivers, including the effects of mining and resource extraction level, etc., then this is something that eco restoration decade, and you can try to tap funding in selecting some of the mined areas for long-term studies, how species can come up and what kind of species can be promoted as a community level approach for promoting biodiversity. The second, and also this is one such approach, there are monitoring work, University of Edinburgh funds, SECO project, and 1 hour 50 minutes funding given. 1 hour 50 minutes. They have been mailing for about 3 months or so, ever since we met in Zurich, the Dry Forest group. And everything set, I told directly it can be reimbursed to students. They said we can be made for them. And I have everything readied and then scanned and mailed to them. 1 hour and 15 minutes, the reply came. And that was something remarkable. And these kind of supports, University of Edinburgh, Scotland, the SECO project will be operative till 2024. So that could be tapped for such small efforts of students. DSc projects, MSc projects, short-term supports of intermediate time for the research scholars. Second important thing is community level studies on phenology with the structural aspects studied, different communities and strata, how they are effectively utilized by faunal communities as separate projects, phenological observations for 3 years or so, 1 to 3 years or so, pollination systems and seed dispersal systems. We need not have to take 10 years like the Malaysian forest Japanese team study, but at least for 3 years of PhD programs by 2 or 3 group on one on pollination and other seed dispersal systems. And that way, through that, behavioral ecology of a couple of species, etc., could be covered in that. Comparing across forest types as part of in-depth forest functional ecology study in better understanding plant-animal interactions, to analyze the complex web of species interactions. Such studies are very limited because we have separate botany zoology departments and our expertise are different. So this is an opportunity under this new national education policy. We can have certain projects and have ready in your box, basket of projects, small projects. When they approach ready-made, you will have something to suggest, saving a lot of time in the process and generating valuable data. Then intensive studies on the species biology of our population study of resourceful species like red sanders we have been talking about, n number of species, both plant and animal side, and how far they are ecologically and economically valuable, and what is the extent of resource extraction that are aimed, and to what extent through self-help groups and other means and modes, the science society interface can be taken care in this exercise. And this part of it, as Professor Dr. Mathur was mentioning about, so it's important that parallelly we have a couple of teams working on specially defined focal programs so that over a period of time, we have a better understanding of a couple of forest systems and biotic groups. So this is the briefing that I wanted to make. I wanted to confine before 4 o'clock. So since we are running short of time, if there are any queries or clarifications, either today or on the second during the field trip in person, students are welcome to interact. And the other important message to the ecological community is that we need really valuable quality data to be generated, and over a period of time when generated, it will be very valuable to science. India needs it. Indian manpower is very, very potential in terms of, but they need good trigger and encouragement. So this is what I sincerely hope that ICERs would provide for the betterment of the institution's growth. My best wishes to the team of researchers in this venture. Good luck to all. Any questions or clarifications, I'll be happy to respond to.
Thank you, sir.
And scholars, my specific submission is please develop skill parallelly in terms of field work, in terms of species recognition skill, which has been diminishing, and in terms of your core ability to analyze data, and last, very important, scientific communication. Scientific communication, writing up into papers, publishing it, and then distilling that and providing to the end users of forest department and other stakeholders distilled from your research for the societal benefit. This is where we need to better understand biodiversity science and conservation sciences stemming from basic biological and ecological knowledge. So.
Thank you. Are there any questions? If not, we'll break for tea. And thank you, sir. It was really great to see in person with data how long-term studies can change our understanding of landscapes.
Thank you,
So,
Nandini, et al.
Thank you.
Thank you, colleagues.
We can break for tea and come back in 15 minutes. We'll be back at 4:15. Yeah.